In this video, we will discuss about tumor-associated macrophages' role in this tumor development, how they affect the tumor development. So for this, first they check the trajectory analysis of different phenotype or different lineage of tumor-associated macrophages. This is the M0-like tumor-associated macrophages. This is M1-like tumor-associated macrophages. It is more dominant towards the right-hand side, and this is M2-like tumor-associated macrophages, which is more dominant towards the left-hand side. And when they checked it in the different conditions in the wild type, they are able to see both the populations, that is M1-like macrophages and M2-like macrophages. But for this chemogenetic inhibition conditions, this M1-like macrophages dominate. And similarly for the case where this Calca gene knockout is used, that is by knocking out this, by preventing the CGRP protein formations, in this case also they are able to see M1-like macrophages are more dominant. And these things they have further quantified at different conditions. So for this chemogenetic inhibitions using HF4 DGI and 3 different cell system, they are able to see decrease in this tumor-associated macrophages. And they checked it for the reverse model also, where they promote the tumors using regional injection of the CGRP. Here they are able to see an increase in the tumor-associated macrophages compared to the control case. That is, in one case where they don't use any chemogenetic inhibitions, in the other case they use chemogenetic inhibitions. So they see in both the cases they are able to see increase in this TAM populations. This chemogenetic inhibitions does not cause the suppression of TAM here in this case. Afterwards, they checked it for one more antibody, this immunocheckpoint inhibitors that is PD1, and they inhibit this PD1 antibodies using or cap these PD1 antibodies using this anti-PD1 antibodies, and in this case also they are able to see a decrease in the tumor suppressions as either in the presence of chemogenetic inhibitions or even if the chemogenetic inhibitions are not present. With chemogenetic inhibitions, the suppression of TAM is statistically significant compared to the control case. Thereafter, they decided to use a model where they wanted to check the tumor-associated macrophages in the recipient mice. So the experiment goes through that first the wild-type donor is treated with either with the control or with this HF4 DGI antibodies. Then this tumor-derived lymph node is taken out, and from that the CD45 immune cells will be sorted. And these immune cells are now injected into the recipient mice that already have this I-type DTR EGFP recipients, and in this case the immune cells are this TDLN is been removed. That is why it is lymphadenectomized. And at the same time, this ITGM will also prevent these myeloid cells. And so in this case, the macrophages that are formed are EGFP positive. So when they checked it in the flow cytometry, what they are able to see in this recipient mice model, and there is a suppression in these tumor-associated macrophages in the chemogenetic inhibition conditions, either when they directly inject the cells or they inject the condition media. The condition media results are in the panel G. In this case also they are able to see a decrease in the tumor-associated macrophages. And when they check it by knocking down this Calca gene that prevent the CGRP formations, in this case also the tumor-associated macrophages signal goes down. And thereafter they use the commercial FDA approved drug, that is Rimegepant. In this case, and for 3 different cell system, they are able to see the TAM goes down as expected. and afterwards they checked it for the reverse model, where they promote the tumor progression using this catch, that is by using this catch which is optogenetically regulated, and it will increase the concentration of calcium in the intercellular part. So this also this will results in the increase in the tumor growth, and that also and that is related to the increase in this TAM that can be seen here. The TAM signal goes up. So this in all together indicates that TAM is directly related to the tumor development. If it goes down, then it will cause the tumor suppression. If it goes up, then it will cause the tumor progressions. This is what they conclude from this part of the figure, from these panels that are presented in this figure. That's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.